Now, let's move on to the other topic of today, which is part 3 of the Stefan Lanka experiments. And here I'm going to share the screen. I think this is probably the best I can do. Maybe make it bigger. There, I did it. Okay, so the title of this paper, and I'm going to basically go through and read some of it, the highlights, and then try to explain it in simple language. So the title is The Structure Analysis of Sequence Data in Virology, an Elementary Approach Using SARS-CoV-2 as an Example. And the author was a mathematician from Hamburg who would like to remain unknown. But I can tell you, I've spoken to the fellow, and he does exist, and he has probably good reasons why he doesn't want to be publicly known at this point. And so let's start with the abstract. So again, I'm going to read it. I'll read a paragraph and then explain what this is. So an abstract is essentially the summary of the paper. And he says, de novo metatranscriptomic sequencing or whole genome sequencing are accepted methods in virology for the detection of claimed pathogenic viruses. In this process, no virus particles, which are called virions, are detected, and in the sense of the word isolation, isolated and biochemically characterized. In the case of SARS-CoV-2, total RNA is often extracted from patient samples, for example, bronchoalveolar fluid, or throat-nose swabs, and sequenced. Notably, there is no evidence that the RNA fragments used to calculate viral genomes are of viral origin. So we all know this. There's two ways of sequencing a genome. One is if you have already sequenced it before, or if you have never sequenced it, in other words, if you're finding a new genome, that's de novo, a new. And so as he says, this is done without ever having an actual sample of the virus. And there's no scientific evidence showing that the fragments, in this case RNA, that are used to calculate this viral genome actually come from any virus. So those are the important points. Next paragraph. We therefore examined the publication, a new coronavirus associated with human respiratory disease in China, and the associated published sequence data with project that number dated there for the original gene sequence proposal for SARS-CoV-2, and there you see the GenBank number. A repeat of the de novo assembly with megahit showed that the published results could not be reproduced. We may have detected ribosomal RNA, ribonucleic acids of human origin, contrary to what was reported in the paper. Further analysis provided evidence for possible non-specific amplification of reads during PCR confirmation and determination of genomic termini not associated with SARS-CoV-2. So the reason we picked this paper is because this is the paper, not one of the papers, the paper that first sequenced this so-called virus, which was then called SARS-CoV-2. Once you have one published genome, all the following genomes use this published genome as the template. So if you can prove that this genome is fraudulent, then obviously every subsequent genome or every subsequent primer, which is the basis of the PCR test, is obviously fraudulent because they're all based on this template. And so that was why it was important to do this paper and this genome because they're the ones who said, we didn't have any template. This was a de novo, a new assembly, and they used MegaHit as the software that assembles the genome. And the first result was we could not get the same result. And obviously, it's a fundamental tenet in science that if you cannot reproduce the experiment and get the same results, there's something wrong. We also think that we've detected RNA, and they call it ribosomal. I question that, but let's not worry about that right now, that are actually of human origin, contrary to what they reported in the paper. So those are the important parts of that. Let's move on. Here we go to the introduction of the paper. And now I'm reading from this first paragraph of the introduction, which says, to construct viral genome sequences, nucleic acids, RNA or DNA, are isolated from various nucleic acid sources such as BALF, nasopharyngeal swabs, cell culture components, or cell cultural supernatants, as well as from human and animal samples and sequence. In this process, the nucleic acids obtained are not exclusively from a previously isolated virus particle, i.e., they're not separated from everything else, but often from the entire sample. Thus, the origin of the nucleic acid fragments used to calculate the genome is a priori unclear. So this is just a restatement that whenever they do sequencing of genomes, either RNA or DNA, they can be from cultures, the supernatant, that's the fluid, they can be from humans, they can be from animals, they can be from your lungs, they can be from your nose, but in no case have we proven that they're actually only from a virus. And as he says, this is very problematic because how do you know that these sequences don't come from other sources? Okay, so we get into that a little later. And now I'm reading this here, this paragraph on page 3. No pathogenic viral particle uniquely associated with MN90, et cetera. That is the sequence that they found. That is the genome that was published as the first SARS-CoV genome, was identified and biochemically characterized from the patient sample. Let me stop there. In other words, no, there is no example of this whole genome in the BALF fluid of the patient. It was not found in the fluid of the patient. That should be clear. Just like you cannot find the virus particle in the solution, you cannot find the whole genome. Rather, the total RNA was extracted and processed from the patient's bronchial fluid. Evidence is lacking that only viral nucleic acids were used to construct the claimed genome for SARS-CoV-2. Further, with respect to the construction of the claimed viral genome strand, no results of possible control experiments have been published. This is equally true for all other reference sequences considered in the present work. Those are other virus sequences. In the case of SARS-CoV-2, an obvious control would be that the claimed viral genome cannot be assembled from unsuspected RNA sources of human or even other origin. In other words, here he's saying, well, if you're claiming that this mess of RNA that you don't know where it's coming from contains in these little pieces the sequence that we call a virus, then you need to do a control, which is to take a sample that say only is from humans and do the same process and find this, there shouldn't be any SARS-CoV-2 genome in there, obviously. And they didn't do that, which means they didn't control for their experiment. So that ends up meaning they have no way of knowing the origin of these sequences because they didn't bother to do a control that would find that out. Okay. Next one. This is a mouthful, but here we go. At this point, the contig is the assembled genome with the identification, you see the number, with which, 1,400,000 sequences are associated, and thus about 5% of the remaining 26 million sequences should be discussed in detail. So in other words, they took, and here we go back, they took the original total of 56 million little pieces and they threw out 26 or so, the 30 million of them approximately, and said, we're going to throw these 30 million out and be left with these 26 million. So why did they throw those out? A large portion of the sequences were overwritten by the authors with N for unknown and therefore filtered out by the programming software. This is to be regarded as problematic in the same sense of scientific whatever, since not all the steps can be reproduced or retraced. In other words, he couldn't retrace why or on what scientific basis these 30 million reads were actually removed. And so there's no way to confirm that this whole thing is actually scientifically valid. So let's get back into this. He says alignment with the nucleotide database on this date, showing a high match with Homo sapien RNA, and with ribosomal and pre-ribosomal DNA. This observation contradicts the claim in the paper that ribosomal RNA depletion was performed and human sequence reads were filtered using the human reference genome. Now, what this means is they said that the reason they got rid of these 30,000 and kept these 30 million and kept these 26 million was because we knew which ones were human and which ones weren't, or at least the program knew. But here he's actually saying these sequences weren't even published until way after this sequence library was made. And then when you go back and do it, you show a high correlation with known human RNA sequences. All this is to say they have no scientific basis for this trimming or construction of what they call the RNA library. It is not based on these ones we know are viral and these ones we know are human. There is no basis for saying such a conclusion. Okay. I'm going to skip the methods, although he goes through in very gory detail how you get all these reads and how you construct the genome and all this. You can read that yourself. It's pretty dense. And then we get to what his conclusions were. So first, we repeated the contig generation. Contig is putting together the sequence into contiguous, a longer contiguous sequence with megahit, that's the program, using the available sequence data and obtain significantly different results compared to the representations. In particular, we were unable to reproduce the longest contig with a length of 30,474 nucleotides, which according to the paper comprised the entire viral genome and acted as a basis for the primer design. In other words, they said this out of the million or so contigs, strands that the computer was able to generate, they picked the longest one based on an arbitrary reasoning. In other words, they said, well, that was the longest one. That must be the genome. It's 30,000, et cetera, base pairs. But he was unable to get that 30,000 genome. He got close, 29,000, but not 30,000. In other words, you cannot reproduce creating the sequence, which then they called the genome. So he says, consequently, the published sequence data cannot be the original short reads used for the contig generation. In other words, you can't get that result from their little pieces. So this is to be regarded as extremely problematic. I would say that's an understatement in the context of scientific publications, since in this way, it is no longer possible to verify the results. The possibility to verify published scientific hypotheses is the essence of a living science. In other words, if you just use their little pieces, you can never come up, even their own software will not come up with the actual genome that they made, which it begs the question, where did they get this from? And then he says in the next paragraph, contrary to what was reported in the paper, we found contigs with high coverage associated with human origin of RNA. Thus, it is possible that not all human-associated nucleic acids were eliminated in the construction of SARS-CoV-2. Further, no evidence in the presence of viral nucleic acids in the patient sample was provided, and consequently, there is a possibility that human or non-viral nucleic acid fragments were used to construct the claimed viral sequence to a significant extent without detection. And this possibility would have to be excluded by control experiments, which they didn't do. In other words, again, he's saying that there's no way to know whether some of the genome that they're calling the original and the blueprint for making the PCR test and the primers and all subsequent genomes didn't have a significant amount of human or non-viral fragments in it, in which case it is clearly not the genome of any virus. And then I'll finish with this paragraph here. In addition to attempting to replicate the assembly published in one with the published sequence reads, in other words, we tried to reconstruct it with the data they gave and we couldn't do it. We considered a simple approach for analyzing the internal structure of large data sets of short sequence reads. With the sequence data at hand, we were able to compute consensus sequences for reference genomes for HIV and hepatitis, with higher goodness than for those for reference sequences we considered associated with the coronavirus. So what does that mean? It means when they took the sequences that were in this published paper, and again, you can see these sequences are here. They go through all the sequences, and in the materials and method, if you're really a glutton for punishment, you can see exactly how they did their mathematics. You can see all the sequences, and what they're saying is, if we took that sequence data, we found the HIV virus that was a better match than coronaviruses. The coronavirus actually only matched 89%, but when we tried to sequence it and used a HIV as the template, we got a better match for HIV and a hepatitis, as they say, with higher goodness, showing that either there was also HIV in this person's lung fluid and hepatitis delta virus, or more likely, the whole thing is a bunch of nonsense. Thus, we were able to substantiate our hypothesis that the claimed viral genome sequences are misinterpretations in the sense that they have been or are being constructed unnoticed from non-viral nucleic acid fragments. In particular, our results underscore the urgent need to perform appropriate control experiments for each suspected pathogenic virus sequence. An obvious protocol would be to attempt assembly of the genome sequence from corresponding non-suspect samples using identical protocols. In other words, if you think that this is the genome, you absolutely need to run the same sequencing process through somebody who's sick or well, or something from somebody who can't possibly have HIV or hepatitis or COVID, and see if you can get the same genome, because even though we haven't proved it yet, we think that will be the case. And if that's true, which we seem to have very strong evidence, at least with HIV and hepatitis delta virus, that that is the case, then the whole thing becomes a house of cards, which we know it to be. And I think that pretty much covers what we need to know. And we will be doing more of these sequencing and trying to actually get this more firmed up over the next weeks, months, and maybe years.